Nanoparticles in cancer therapy. What is nanoparticles in cancer therapy? Nanoparticles in cancer therapy is the tiny particles smaller than a human cell, which is designed to treat cancer more effectively, precisely, and safely. They also act as carriers to deliver medicines directly to cancer cells while avoiding kill the healthy cells. How to form the nanoparticles? To form nanoparticles have two ways. First, top-down approach. Second is bottom-up approach. What is the top-down approach? Top-down approach is break down the bulk materials into the nanostructures. Why? For the bottom-up approach, we assemble the atoms or molecules into the nanostructures. For the top-down approach, there are two methods used this way. First, physical synthesis and chemical synthesis. Why? For the bottom-up approach, also have two methods used this way. First is the chemical synthesis, second is the biological synthesis. How the synthesis operates to produce nanoparticles? First, through chemical synthesis. It is a process using chemical reactions to produce nanoparticles. For example, soldier methods, hydrothermal, chemical vapor synthesis. So, for example, soldier methods, how it operates. First, we dissolve the metal alkoxide, which is as the precursor solutions, with a solvent such as water. Here, we produce the solution formations. This process we call hydrolysis. Next, we allow the condensation reactions occur to form a gel-like structure. Then, we dry and heat the gel to form nanoparticles. For the physical synthesis, it a process produce nanoparticles using a mechanical process. For example, laser ablation methods, ultrasonication methods, electrospinning methods. For the image that I choose is a laser ablation methods. It is a simple and effective way to synthesize nanoparticles using high power laser energy. How it operates? First, the high laser beam will focus on the target material, which is solid material, such as metal or ceramics, and we place it in a liquid medium. Next, the laser will hit the target surface that cause it to vaporize or eject the material into surrounding medium. The ejected materials will cool and condense into nanoparticles within the liquid. And the last one, we separate and purify the nanoparticles from the liquid for further use. For the biological synthesis, it is a process produce nanoparticles using biological materials, such as plants and microorganisms that consist of algae, bacteria, fungi, viruses. So, for the biological synthesis method, how it operates? First, we will extract the plant extract or we use the microbial cultures. Then, we mix the biological extracts with metal salt solution, for example, silver nitrate. Then, we stir the solution at the optimum conditions. Here, the chemical reduction of metal ions will occur, and then will generate the nanoparticles grow. So, we filter the solution to obtain nanoparticles. This is a general image for the components of nanoparticles. We can see the size of nanoparticles is within the range between 1 and 100 nanometer. For the material, we can see there are multiple types of core material that we can use. And for the shapes, there are different shapes of nanoparticle that we can apply, and also there are variety ways of the surface modification can use. Four main components of nanoparticle. First, core material as the structural base of the nanoparticle, which determines drug loading capacity and release kinetics. For example, polymeric cores, biodegradable polymers like PLGA, chitosan, lipid cores, solid lipid nanoparticles, metallic cores, gold, silver, iron oxide, inorganic cores, silica, calcium phosphate. Second is drug payload. For the active pharmaceutical ingredients, for example, chemotherapeutic drugs, doxorubicin, paclitaxel, gene therapy agents, siRNA, plasmid DNA to silence oncogenes, immunomodulators, checkpoint inhibitors, cytokines, vaccines. Third is a phase modification to enhance targeting biocompatibility and circulation time. For example, targeting ligands, antibodies, peptides, or aptamers that bind to specific tumor receptors, stimuli responsive coatings, drug release when respond to heat, light, pH, or enzymes. And fourth is stabilizer or surfactants, which maintain nanoparticle stability, prevent aggregation, and ensure uniform size distributions. For example, surfactants, Twin 80, and polymers is PEG, polyethylene glycol. The mechanism of nanoparticles delivery in cancer therapy. It means how we deliver the nanoparticles into our cell body in the cancer treatment. There are 4 mechanisms to deliver nanoparticles. First is a passive targeting. The passive targeting, it enhances the permeation and retention effects, PPR, which is the nanoparticles passively accumulate in tumors due to leaky vasculature and poor lymphatic drainage. Second mechanism is active targeting. The ligands on the nanoparticle surface will bind to the specific tumor cell receptors, such as folate receptor or HER2 receptors. Third mechanism is control and stimuli responsive release. The drug will release when triggered by the tumor-specific conditions. For example, during condition of the low pH or high enzyme levels. And the last one is the combination therapy. The nanoparticle can co-deliver drugs, genes, or adjuvants to achieve synergistic effects. It means the nanoparticle can deliver 2 or more therapeutic agents in a single nanocarrier system. There are lots of benefits of nanoparticles in cancer therapy, such as will enhance the targeted delivery, can use as the theranostic applications, which is nanoparticles can combine the therapeutics and diagnostic capabilities, can control and sustain the drug release, can overcome the drug resistance and will reduce the side effects and improve the drug solubility and stability. There are some cancer diseases that can use nanoparticles in the treatment, such as brain cancer, esophagus cancer, the lung cancer, blood cancer, liver cancer, gastric cancer, pancreatic cancer, colon cancer, bladder cancer. The future and innovations of nanoparticles in cancer therapy. Smart nanoparticles. These nanoparticles can deliver therapeutic agents directly to the cancer cell without damage to healthy cells. Nanoparticles in space medicines. It is developing the nanoparticle injection which aim at to reduce the effect of high radiation exposure during prolonged space travel, which is potentially prevent the cancer and other health issues. For the nanomachines against drug-resistant cancers, it is a development of molecular machines that disrupt the cancer cells by using the specific light wavelength. This method to fight the drug-resistant cancers. For the gene and RNA delivery, nanoparticles play a critical role in delivering gene editing tools. For example, CRISPR-Cas9 or siRNA, which is to target oncogenes or repair tumor suppressor genes. And for the magnetic nanopharmaceuticals, is the nanoparticles combine specific antibodies with magnetic materials to precisely target the cancer cells. In conclusions, nanoparticles offer a promising pathway towards personalized and more effective cancer treatments, revolutionizing the fight against this complex disease.